Okay, yeah, Lawrence already announced my title, Making AI Fit for the Life Sciences. And I'm really happy that Lawrence did the same as I'm going to do, because for me, AI is machine learning and deep learning, so I just use the terms interchangeably. So if you expect some other AI, sorry, not today, but maybe in a different seminar series issue or speaker series issue. So first, some words on myself, if this is working, but it's not. There we go. That's a little timeline of where I came from, what my background is, and where I am now. So I started off studying mathematics at University in Berlin, then did my PhD in theoretical biophysics. Now you might say, oh, what a switch. But the reason was I was always interested in biology and biological mechanisms in life sciences, how life is working. I even contemplated studying medicine, but decided for math in the end because I also loved math. Good idea, because I also do like what I'm doing right now. So I advanced doing my PhD in theoretical biophysics at the Magdeburg Center for Molecular Medicine here in Berlin. Was a postdoctoral researcher there and did further modeling. And then went to the Luxembourg Institute of Health, where I did a postdoc starting to work with networks. And before I joined Free University in 2023, I was a senior researcher, which is a group leader at the Hasso-Plattner Institute for Digital Engineering, which is a computer science institute in Potsdam, close by, where I ventured into computer sciences more. And you see already connecting the three. Now I am an assistant professor for data integration in the life sciences at Free University Berlin. And this is how Free University is looking like, or at least a little bit. So this is the Silberlaube, you might know, and this is actually the campus where I'm sitting. The office is here in this border close to the Zuse Institute Berlin that has a lot of computational power, you might know. It's quite idyllic. There are a lot of trees, which is also nice because it's fitting to my lab. Well, my name, if you know a bit of German, you know why. And it's in the Department of Mathematics and Computer Sciences. And why is now data integration so important, and why do we need it in the life sciences? And when I say life sciences, what I mean most of the times is health and disease, because it's so important how things work, what's going on there. So why is it so important? The human body has a lot of cells, so 10 to the power of 13 cells approximately. And in each cell, there are even more protein molecules, if you want, and many more other types of molecules as well. In addition to this complexity that is down on the molecular level, every of us 8 billion people, around 8 billion people, is also different and lives in a different environment and is a slightly bit different, or even a lot of different. So there's a huge amount of variety for us that we need to tackle, that we need to find, where we need to solve the riddle of this complexity. We do this by data, by measurements. So we try to assess how life is working by measuring life processes, cellular processes, assessing electronic health records maybe. And each of these data stem from different views, and thus they form different layers. So you have different layers of data, and each layer has errors, comes with its biases, is only a very single reduced perspective of the overall complex system, contains missing values. So it's difficult to get the right conclusions from just one little perspective. So what we need is integrating these data. So find and leverage connections between them in order to enable correct conclusions. So this is what my group is about. So called data integration in the life sciences. We're working with multi-layered data here of different scales. I will come to that and talk about it a bit more in detail, what we're focusing on also in this talk. And as you might have guessed from my background, we are also combining different methods. And these different methods are nurtured from my background. So I started off with dynamic models in theoretical biophysics, ventured into working with networks at Luxembourg Institute of Health, and then started to combine the two with machine learning at the Hasso-Plattner Institute for Digital Engineering later. And now I'm combining the three, which is an exciting, let's say, place and time to be to combine these different approaches. And I will tell you part of that today, where we combine dynamical models and machine learning in a short while. What is the life scientific data that we're focusing on? It's molecular data most of the time. So this is a huge cell. You have here the nucleus and the cell membrane. And I talked about these different types of molecules. Proteins is one of the type of molecules, and there are different other types. And if you measure these different types of molecules, you would have omics data. And these omics data are in tables. And I already talked to Lorenz, who was very fascinated about his community data set that are images. Well, we do not work with images, and the talk that I'm giving you today is not focusing on images at all. So the focus and there will be images, but the focus is not on image data, but on tabular data. And this is what our group is also dealing with. Then we are also considering drug properties. This will not play a role so much. So if you answered the social question and were interested in how to use ML for drug design, also not today, sorry. So if you speculated on that, but we're also dealing with time series on different scales. So life is made up of different scales. I talked about the molecular level. We have cells, tissue, organs. People interact, patients. We have populations that do interact and give rise to different time series. And this is of interest to us as well. So this type of data. Okay, that's a type of data. Now, why machine learning there, or why AI? And as you probably all know, what is machine learning at all? Machine learning excels at finding pattern in any data, including life sciences. So what is machine learning? Short recap. We want to find a map from an input to an output. We have examples for that, training data. We apply our machine learning pipeline where we train on this data. We have our hypothesis that we learn. Maybe we validate our final hypothesis, and then we have our solution or approximated function that is giving us from an input and output. And there are different flavors of that. Random forests, neural networks, even more complex architectures like graph neural networks, and so on. And I told you that it excels at finding patterns, also in the life sciences, and proof of that is that it's also strongly used in those papers that are relevant for our field, for the field of life sciences. PubMed is one of the resources where we find those papers. And if we check PubMed results for the 3 big journals, Cell, Nature, Science, that's the high-class golden standard journals in the field. And you see it's increasing over time already. I did that in the end of 2024. So sorry, it's not the very recent data. And if you go for more journals than just Cell, Nature, Science, the numbers are as you would expect. So it's not 34, but more in the realms of 20,000-ish publications published in those years. So it's really advancing. You use machine learning and deep learning, especially in the life sciences. Images would be there as well, so I didn't exclude that, sorry. Okay, now, what is the questions that we solve when we deal with this life science data? Actually, also with images most of the times. And there are a wide variety of different questions that you have, but everything basically boils down to one question. How does life work? That's what we aim to find out. Why do we want to find this out? Well, we want to exploit, we want to forecast, manipulate, maybe use mechanisms that appear in nature for our engineering. We want to go back to health and get healthier. We want to find out what is wrong in order to find which intervention could make us good again, could make us well again, especially in health. Now, solving this basic life scientific question with machine learning and deep learning gives also rise to problems when using this. And the problems, especially of the data that I described to you, are two of the problems are those. And one problem is small data. You might not think that there's small data, but if you do not have images, where you have plenty of data usually, all the tabular data, the molecular measurements are rather small. And the reasons for this in life sciences on health is that if you want to measure something, the sample is being spent, right? So like you dissolve the cell membrane and the cell is then gone basically after you have measured it. And you need an amount of really tissue in order to measure how many molecules are there, at least if you want to go in depth and have all those molecules. Then what comes into play is this very wide variety that I described before. Many, many different molecules give rise to a wide variety, high complexity of the possible space of options, what is out there. So you might have a lot of examples, but just the space of possibilities is so big that your sample is still small compared to what's possible. You can have rare diseases where you have even only 1 in a million or 1 in 100 million patients that have this certain disease. So finding someone who's similar to do statistical stuff like modeling, training on this example, very tough in machine learning. And then in addition comes measurement techniques tend to be expensive. So not all, like genomics is affordable-ish, but as soon as you want to go more in detail, measure proteins, measure even change proteins like signaling molecules, it's very expensive. So this is also reducing your amount of data that you have. What is a second problem apart from small data? Second problem is transparency and trust. So if you have a machine learning model, it's usually considered as a black box. And in order to be able to use these AI models in health and clinical settings, it's required to have a certain transparency, to have people trusting in the model because lives are at stake, right? If a decision depends on the decision of a machine learning model, you would only do this, you would only allow this if you trust this model, if you're sure that it's doing the right thing. So it's highly regulated in clinical settings and you have a high responsibility that is associated. I today want to describe you or brought you 2 stories that are addressing these 2 problems, small data and transparency and trust, what we are working on. It's not the only solutions. These are also not the only problems. Of course, they're more in-depth, and I will stay on a very methodological level and start with small data. So small data means in our pipeline where we try and fit our machine learning model, this part is interrupted or, let's say, altered, reduced. You have a reduced amount of training data. How can you overcome this? You could try and measure more. Yeah, okay. But you could also try and include other knowledge, prior knowledge. And this is called informed machine learning. And what we use in order to inform our machine learning models is use information from dynamical models. So going for this interaction or for this combination of machine learning with dynamical models. Why dynamical models and what do I mean when I say dynamical models? Dynamical models are descriptions of time-varying processes most of the times. They're mathematical representations, which is great because if you want to include them into machine learning that you know is very formalized, is mathematical, we need a mathematical representation. It can or they can capture intricate biomolecular and knowledge curated by researchers. So they have spent hundreds of years researching single mechanisms within a cell, for example, measuring in depth and establishing how entities interact and how they can be described mathematically, these interactions. That is one example that is not so intricate and not so complex, but it's useful. And it's a model that you might know. It's a susceptible, infected, recovered model where you model infection dynamics. So you have a susceptible population, infected and recovered or removed population, and then you can solve this for certain initial conditions and kinetic parameters, and you have this time series or time course where you see how the infected have this dip here and the susceptible are decreasing over time. Now, how do we get these dynamical models? Well, there are databases for that. And one of the database that I would like to point out that is possible to use is the so-called biomodels database. It's a database that is hosted from the EAMBL, from the EBI, European Institute of Biology. And it's hosting curated and not so curated models in a standardized format or close to standardized format, SBML, of very different organisms. And actually the model type that they use most or that is represented most is ordinary differential equation models. So a lot of systems biology knowledge, a lot of domain knowledge on life sciences, on biology, on different cellular processes or at all processes are captured in these models. So there's a huge potential, let's put it like that, of prior information. Now, what processes could be represented? I just brought some descriptions. I will not read it through. It's just on every scale, you can find models of different processes that can be represented by those models. And in addition, you can also have like models that span populations, like ecological networks where you have predator-prey systems that are described. Now, how do you use them in machine learning? And one issue that we faced when we thought about wouldn't it be grand to combine this dynamical modeling with machine learning is that they tend to be different communities, right? You have the community of modelers, of those people establishing ordinary differential equation models of intricate pathways that are very complex. And you have the other community of machine learning people training their models. And these communities are not the same, let's put it like that. So it was difficult, or it requires different expertise to combine the two. So what we did was to set out and establish a toolbox that makes it easier to combine the knowledge from ordinary differential equation models with machine learning. And for that, a group of students at the Hasso-Plattner Institute, Max and Benedikt, Lukas and Julian, under the supervision also of Bernard Renard of his group, that was back when I was still there at Hasso-Plattner Institute, started and created this toolbox, this Python-based toolbox, where you can put in the upper part, or where you can create a simulation with ordinary differential equation models in the upper part, and here in the lower part, the machine learning pipeline, and everything is in a single toolbox. So you give the pipeline via the simulation configuration file a model. It would solve the ODE, add some noise to make it look more realistic. It can also be captured. You can take the synthetic data off the system if you want, and or combine it then with real-world data to train your machine learning model and do your predictions in the end. Sorry for that. It's compatible with scikit-learn, TensorFlow, PyTorch, Geometric, whatever you want to use as machine learning model. And because they're at the Hasso-Plattner Institute, which is a digital engineering institute, they were very keen on making their code nice. So we have 100% test coverage, which is great and not very useful. It's very useful. Sorry for that. This was it's not very common in those research-based packages. So give it a try if you want. Now, how can you use that? What can you do with it? And one drawback of this model or of the SimbaML is that it's only there for creating synthetic data of time series. So what you can do is you can improve time series forecasting because the synthetic data that you create, or the simulated data that you create, are time series. And an example of those time series would be if you have here the signaling cascade. It's MAPK cascade, very common signaling pathway. Don't worry, I will not bore you with any details here. It's just the cell gets a signal and then it's being processed, let's put it like that, in the cell. And the final output would be a fully activated intermediate signaling molecule, doubly phosphorylated MAPK. That's just that. That's why the MAPK pathway. And what you can try and do and predict, or what a task could be, is you have some data points measured of this activated MAPK and you want to know what the next 3 time steps are. This would be a time series forecasting problem that you can try and, let's say, process with our SimbaML. Or you could also try and find infection counts right from the start. So these are 2 different prediction tasks that you could imagine here. And now, what could you do? And I brought you 2 examples of how you could use SimbaML. And one example is, assume you want to do this prediction task of predicting how much signaling molecule you have 3 time steps in the future. How much data do you need? And what would be the best machine learning model? And how much data do you need? It's a really crucial question for us as machine learners, right? Or as machine learning people. So, of course, we say we need as much data as we can get, but that's costly, especially if you have life science data. So, better know beforehand how much you need in order to get a certain accuracy. And this is what we did here. So, the prediction task is forecasting of the central signaling molecule, and we used the SimbaML to try and find out how much data we need and in which setting, which accuracy do we get for which type of models. It's a very small study. So what we did, or what we need for that, is we need an ordinary differential equation model of this MAPK signaling pathway. That's an example for that. It's from 2007. It's, as you see, a bit less complex than the whole signaling pathway, but that's fine. It's a system of 8 equations here, where the key signaling molecules are being modeled over time. And these are examples here of the time series that we see. And then we simulated some synthetic time series, only one-dimensional, so only the activated signaling molecule, the time series over that. And you see it's a quite boring dynamics. It's just a sigmoid, but it's also only a case study, could make it more complex. For example, predicting from multiple molecules this time series or so. But we did it for this example. And now we trained different machine learning models for different numbers of time series. And this is the results that we have. So here we trained on 5 time series, on 10, 100, 1,000, 10,000, and 100,000. And we trained 5 different ML models on that time series forecasting task. And, well, it probably depends on what you want. But probably you do not want to measure 100,000 time series, but it's for the sake of, say, completeness, we still did that. Because what you see is that for your dense neural network that is here in green, predictions are, this is RMSE, the smaller the better, so the error. The dense neural network is only getting decent prediction comparable to the machine learning model as soon as you have a lot of time series. And this is expected because a neural network, well, needs a lot of data. Other models do not need so much data to get decent predictions. For example, the random forest. It's known to perform quite well, usually, most of the times. And also here, the random forest in red is performing really good from the beginning. Even with 5 time series, well, yeah, variation is a bit bigger, but it's performing decently. What we also observe is a linear regressor. So if you assume a linear regression instead of this sigmoid, let's put it like that, the linear model is so the performance near constant, depending on the average performance at least, is near constant, independent of how many time series you have. Well, so if you have very few, you might still go for the linear model. But if you have a little more, there might be also still, but if you have a little more time series, then linear model is just too simple to predict the sigmoid. So you can do better. Okay, so that's a little example that how you could use SimML for assessing which ML model, also how many time series you need in order to get a decent prediction. You have a question? Go ahead. So the question is, what is the difference between implementing the ODE directly into the machine learning model instead of feeding the synthetic data into it? There I didn't feed, so there the idea was just to assess how much data, measured data I need, assuming that the that the time series is behaving as in the model itself. The next example that I'm going to show you is a case where we use the synthetic data to pre-train. And this is a task where I added to the model, and there it goes even further. So there's a different rationale how you can use the prior knowledge and put it into the machine learning model, which is by, for example, putting it into the loss function. This is called physics-informed machine learning as well. And it performs a little differently, and it depends on the case how you do it. We focus here on the case of pre-training with synthetic data, though. So it depends. It can be similar, but there are also differences. Okay, maybe I go on. So, and this is exactly, thanks for the question, this is exactly what we're planning to do, or what we did here. Instead of just assessing how much data you need, you can also use SimML to pre-train on simulated data. And this is what we did in the next step. So we used the ODE to simulate, put it into our machine learning pipeline to get a pre-trained model that is already biased towards the dynamics that we want, and then use the real-world data and only do a slight fine-tuning to get our final prediction model. And this is what we did with this infection scenario here. So we used a susceptible, infected, recovered model that I showed you before already, not so complex, and created synthetic data, including noise, and then tried some infection prediction without in pink or with synthetic data in green. And this figure is a bit complex, so I just walk you through. So it's 3 different experiments actually that we did. And each experiment gets a certain amount of real-world data, and how much real-world data is indicated by this dashed line. So the first experiment is only giving the real-world data up to this point in time. The second experiment is giving it up to this point in time. And the third experiment is giving it up to this point in time. The synthetic data that the model sees is always the same. It's always these time curves here. And what you see is that if you give it really, really little real-world data, the model that did not see any synthetic data is performing a bit better, which is blue is real world, than the model that received the synthetic data. So it's not very successful, you would say. But this trend is changing if you give it a bit more real world data, because then the model that did not see the synthetic data tends to shoot into growing numbers, whereas the other model that saw that in the end there will be the infection numbers will go down or the new case numbers will go down, is adapting to that. And it's even more blatant here, where the model that did not see any synthetic data has never seen a downwards trend that the model with synthetic data has seen. So in this case, it's really, really helpful to use the synthetic data to pre-train your dynamic models, to pre-train your machine learning models. But it's not the case all the time. So next step was we wondered, when is this the case? So what needs the synthetic data characteristics to be, or what is the system characteristics that is required in order to make this simulation-based transfer learning, how we call it, or how it's called, successful? And for that, again, the same students who also wrote SimbaML did their master thesis with us, and not only their master project, and build an optimization and evaluation pipeline for the properties of synthetic data sets that you can use for simulation-based transfer learning. And what they considered were three different factors of the synthetic data set. First, the data set size, so very few time series, or one time series that is close to a naive, not pre-trained model, to many time series. Then we have the dataset diversity, and this is marked by how strongly do you change initial conditions or kinetic parameters when you simulate your dynamical models, and whether and how, which type of noise you introduce. So whether you have measurement noise or environmental noise, and whether it's additive or multiplicative, and how strong the noise is that you use. Then you would do data generation with SimbaML, you would do the simulation-based transfer learning, and also a naive baseline where you do not pre-train. And we also considered the ODE baseline with it. You would apply this and evaluate the differences between the DL and ODE baseline with the simulation-based transfer learning. And we had, of course, also a proof of concept setup. So it's not very complex here. So really proof of concept. So we had three different models. Again, the SIR with the COVID-19 data set, and predator-prey, and two predator-prey systems, actually. So rotifers are small bacteria that feed on algae. So it's a biostat, the big tank, let's say, where it's not tank, it's a big glass bowl, let's put it like that, where these bacteria and algae are living and dying and being eaten. And then we have the famous lynx hare data set, which is really this one in Canada, where you have the snowshoe hare and lynx that are feeding on those hare in Canada from 1830 to 1940 or so. And these are modeled, or this is modeled with a Lotka-Volterra system that is very famous predator-prey system. And this rotifer algae has a slightly adapted Lotka-Volterra system with another food source. So this is the example data. It's oscillatory. They have different properties as well. The SNR is not oscillatory. And we set it up and did our simulation-based transfer learning, compared it to the DL baseline and ODE baseline. And this is one example of the best performing transfer learning and DL baseline and ODE baseline of the lynx hare time series forecasting. And I'll just walk you through. So we give it data or real-world data up to a certain point in time, every of the three models. And then we have our predictions, that is 5 time steps into the future, given the last 5 time steps. And these red traces are those examples that you can create from the data that we have. And this is the simulation-based transfer learning. This is the deep learning baseline, and that's a very simple ODE baseline. So comparisons have to be taken with caution because the ODE baseline is really, really simple. But these can be compared very well. And what you see is that if you optimize the synthetic data that we put, we can really improve our transfer learning. So you see the gray curves here are better fit by this red curves rather than by these blue curves where we didn't pre-train. It's even undershooting, so we even have numbers lower than 0 predicted. So it's improving, at least for this example. And we, of course, did that for the other examples as well and showed here the improvements in percent, and what is boxed in red means that the simulation-based transfer learning is a bit better than the others, or even considerably in some cases, both in terms of mean average, mean absolute error, and 1 minus trend prediction accuracy. So the predicted trend in the time series is very important as well. And these are the optimized characteristics of how many time series you have and what the initial conditions and kinetic parameters, how strongly they are changed. And we also did that for 4 different architectures, ML model architectures. And what you can observe is that if you optimize this simulation-based or the synthetic data, you can really get improved results upon the deep learning baseline. And Julian and Max also set out to assess, of course, the effects of what happens if you have different synthetic dataset sizes, the variability, and the noise. And they made the different multivariate analyses. And we found, of course, interactions between, if you have a big data set, then of course variability is more important, because simply the phenotype is expressed more. So if you sample 1,000 times, the variability is more pronounced than if you sample just once, even if your selection interval is quite big. So these types of things. And actually for the noise, we also found that it can help if the data set is not really fitting to the ODE model itself. Then it's helping to add more error, because then the pre-trained model is looking more like a naive model and you do not wrongly bias it. So I didn't bring you the details here, but overall what we really saw and need is it's good to have a coherence between the data set and the model that you use. If you have the coherence, then also bigger data sets are good, but you need to optimize these synthetic data sets, because if it's not coherent, then probably smaller pre-training, less pronounced pre-training is better. I want to sum this up. So we addressed the small data problem here in life science data with machine learning by informing machine learning with dynamical models. We found that combining machine learning with the knowledge from systems biology by using this data from ODE simulations can really improve predictions, but you have to optimize the synthetic data set characteristics. And it's an example for machine learning interacting with dynamical models. And these are the 2 papers again, so you can check it out. All right, that was problem 1. Now, let's come to problem 2. Transparency and trust. This is what I was talking about earlier. It's important in clinical settings to have transparent and trustworthy machine learning models. Why in clinics? So assume someone is presenting at the doctor and the doctor applies a machine learning model where the dose of the medication is given. And if you have a normal machine learning model just like that, well, the doctor says maybe, oh, it's such a big black box. Can I really trust this prediction? And there are, of course, solutions to that. You as a AI, machine learning, deep learning community probably know what solutions could be. And it's uncertainty estimates and model explanations that have been proposed. So what can you do? You can make this black box more transparent by adding estimates of confidence, of uncertainty of these predictions. So for example, here, instead of having just 20 milligrams, you say, okay, 20 milligrams plus minus 5%, that's my 80% confidence interval. Then probably the trust is better and you know, okay, yeah, 20 milligrams is fine. Model explanations means instead of just saying dosage is 20 milligram full stop, the model is also providing a rationale. Why 20 milligrams? Well, sorry for that. Because some parameter in the blood data set is higher than 10 and the age is smaller than 50. Maybe that this is something the doctor can assess. Is this true? Do I trust the results? Yes or no? And might be happily using it. Now, uncertainty estimates and model explanations it's lying around. There's a lot of methods for that, and we are not developing a novel method for that. That's just illustrating that there are different types of uncertainty. You have the type that is inherent in your data that is called aleatoric uncertainty, and you have the type that you could maybe eradicate if you gather more data that is called epistemic uncertainty. And there are different models to assess these types of uncertainty, like Bayesian neural networks is classical, quite computationally expensive, mixture density networks, and you have also different methods for explanations. You probably know all of that, like here integrated gradients or layer-wise relevance propagation and SHAP values and you know what. So there are many methods for that. But what we set out was to go a step beyond that. So to further increase transparency and trust, beyond just explaining an uncertainty, but explaining uncertainty. And we set out to explain aleatoric uncertainty. And there are methods around to explain epistemic uncertainty, and there are also methods around to explain aleatoric uncertainty, but they are quite bulky, computationally expensive, and you cannot just plug it to your model of choice that you have, so to your neural network that you have. And the rationale, why would you want to explain uncertainty at all in this clinical setting is, well, dear patient, we have this AI tool, we have assessed uncertainty. It does not work in your case. We are too uncertain, sorry. This would be having uncertainty. But why? Why does it not work? And this question is something that is addressed by explaining uncertainty. And for doing that, we propose to go a very straightforward way that is not new, that is being composed of parts that are around even for 20 or 30 years. So, and this is equipping a neural network. If you have a neural network point-based prediction, you equip it with a Gaussian distribution assumption. So instead of just predicting the point, you would predict a distribution, like add a variance output. Let's put it like that. It's easily plugged in, really easy. So if you have the old weights, what you would do is you increase another column of weights that is leading from the last layer, second to last layer to your variance output, and you would then, instead of your normal loss, you would optimize a Gaussian log likelihood loss. And then you can use this output that is giving you an estimate of the variance of the uncertainty and apply any of those explanation methods that you want. For example, Shapley values to get explanations of the uncertainty in your predictions. And we term this variance feature attribution. As I said, it's not new. You have a variance feature that you attribute things to. That's why the name. So we just needed something short for this method. The good thing is that you can really plug it in a neural network that you want. So you pre-train it anyways for your mean, and then you add this variance node. Now, what we did was to benchmark this method against other methods of explaining uncertainty. And for that, we established a synthetic data set here where we have variance features, so uncertainty features and signal features. We have a linear signal, and we model the noise by some polynomial, so it's more complex than just linear. And then we sample from a normal distribution around our signal, but depending on the noise here. We also did it for mixed features, so where features do not only enter into the noise or only to the signal, but also in both. And this is actually an example of how it would be sampled for a one-dimensional synthetic data set where the feature is both entering into the mean and into the noise. We also used 3 real-world data sets on red wine, on something on airplane wings, and on the law school grades. And we compared our variance feature attribution with different explanation methods, integrated gradient, layer-wise relevance propagation, and SHAP, to 2 established methods. One of this is counterfactual latent uncertainty explanations, CLUE, and InfoSHAP, that is based on XGBoost models and training an auxiliary XGBoost on the residuals. Now, first of all, if you think explaining uncertainty is just the same as explaining the mean, that's not the case. So remember, you have these 2 output neurons here now, and you can explain both the mean. Here are the features for that, for the synthetic data set, and you can explain the variance. And what you see is that the top scoring features are different. So you get the mean features are some of those 70 mean features, but for the variance output, very frequently the top performing or top scoring features in SHAP here are really noise features. So it's working quite well. And we also quantified that and set out a benchmark on methods that are established for XAI, but are not so established for uncertainty or explaining uncertainty. First of all, we adapted the relevance rank accuracy and relevance mass accuracy here by just counting. So if we have a ground truth that we have in our synthetic data set, we can compute how many of those top scoring features are overlapping with our top scoring by our prediction. And we can also do this by the rank, so add up the rank of the top features. And we can make this global and local. I do not need to go in detail here, but rather the results. And the results is here comparing our three approaches with the two competitor approaches. And what you would want is that the red features that are significant or that are signifying the noise features are top ranked. And this is what we observe in our three methods, in all methods with variance feature attribution. You do not see it so much in InfoShop, and you see it in CLUI, but only if you go for features with very high uncertainty. So for instances with high uncertainty, 200 instances, averaging over those, CLUI can identify those noise features, but in the other cases, the other competitor methods are not so good. Further, not only accuracy, but also, or let's put it like that, we can do this also on the real-world data sets, but only if we have ground truth. Since we don't have ground truth in the pure real-world data sets, what we did was to introduce some noise features artificially to have a semi-synthetic setting. And then we also mapped here the rank accuracy and the mass accuracy again, and we saw that our VFA is performing really well, and VFA sharp actually the best all the time. Runner-up most of the times is also a second of the VFA methods with integrated gradients, except for the small data set here. So rank-wise, the VFA, the very simple approach, is performing really well. What about faithfulness? Faithfulness, there the idea is that if you perturb features that are relevant, they are not predictive anymore of the real deviation of your result from the prediction. And this is what we assess here. So we want our models itself, our variance predictions, to have a high correlation with the error that is done. So this is the error in the prediction. And we compare it if we alter the top ranked features, and we expect that this correlation is dropping. So if we alter features that are relevant, we expect that the predictiveness is reduced. So more negative is better. And this is actually, we did it on these three data sets. We do not need ground truth here, but we did not do it on the LSAT because on categorical features, this perturbation does not make so much sense. And we saw that, well, here for the smallest data set, InfoShop is best, we'd say, but for the other, the correlation is really being reduced. Last, benchmarking metric, robustness. There we set out to compute this Lipschitz estimate, where you see for certain neighbors if the change in the prediction of the explanation is changing a lot. If the difference between the neighbors is big, and the smaller the better. And you see here also that the three different VFA flavors that are the first three here are performing quite good. The VFA LRP not so much, so there are some instances that are really not good, but for most of the times it's performing well, and also InfoShop is performing quite good. So we saw over all these three different benchmarking metrics, robustness, or accuracy, faithfulness, and robustness, this very simple approach that you can just plug into your neural network is working quite well. We did another prediction on images. So here they are, I promised you. So we did age prediction on IMDb actor images, so not very biological, but also the other data sets were not. So we tried and predict the age, used an existing model for that, applied VFA to it, and these are the explanations of VFA. And what we see is that the explanations are really following those expression lines. So emotions that can interfere with age prediction. So you have wrinkles from emotions can interfere with wrinkles from age. So you see here that the explanations visibly could make sense, could also be relevant for the mean prediction, but they are actually a bit different here. All right, I want to summarize also this part. So we try to improve transparency and trust by explaining uncertainties, so adding an explanation here to this uncertainty setup. And for that, this straightforward approach of equipping a neural network with variance estimations by using this mixed distribution networks is working really well and applying standard explainability methods. We get accurate, faithful, and robust uncertainty explanations for these regression problems. And to summarize, I showed you how to address two different issues for dealing with life scientific questions and problems when using AI. So first, informing ML by simulations, and second, explaining uncertainty with variance feature attribution, for example. And this is moving towards using AI or making it usable in the life sciences, we think. I'd like to acknowledge some people. What I left out today entirely was data integration part. Be welcome to check our homepage here. There are some more projects, not many, but some of them where we do this more in data integration. And now I'm also happy to take your questions, and thank you so much for your attention.
So thank you so much for your presentation. And now Anna will bring you the microphone so that you can ask your question.
Thank you very much. Does this work? Yes. So hi, thanks a lot for the presentation. I had a question on problem 1. And let's say you're in a world with small data or not a lot of data, right? You have 1 to 3 data sets. And then the modelers working on the differential equations will use these data sets to develop their models. The machine learners would also be looking at the same data sets. And I wonder if we could get some kind of information leakage if, let's say, the modelers use the entire data set and the machine learning people split the data set between train and test. I wonder if information could leak if you are in this world of small data.
Yes, I fully agree. If you use exactly the same data set to develop your model that you use your machine learning on, it's not useful. That's just what I have to say to that. Yeah.
Hi, thanks a lot for the presentation. So I'm wondering what's the next step for research and for your group? Are these the main problems you want to tackle in the future too? And how do you see the field evolving? I mean, especially seeing as AI becomes more and more used also in the life sciences.
Yeah. So I think exactly this combination of machine learning with prior information is something where a lot of potential lies. And I showed you here one option to include dynamical models, but there are others. And I think there we're not at the end yet. I think this combination of prior knowledge from dynamical models into machine learning is something that is very valuable, needs to be researched a lot more. And then there are other sources, which is not necessarily dynamical models, but for example, network information. There we are a lot more advanced already, like in graph neural networks and so on. And bringing this to the domain of life sciences is something that is very valuable and where we still need development work, because life sciences is coming with a lot of uncertainty, and you have to you have to assess what the effect of this uncertainty is, also in the prior knowledge. And I think there we need a lot more research, and I think this is a very fruitful way into the future.
Thank you for the presentation. That's really nice. Also from the problem 1, I was really curious about your some of your slides on multi-scale problems, where you have, I don't know, phase transitions between different regimes, molecule versus protein versus.
Phase transitions. Do you mean more complex systems, right? Where this plays a role. So this approach has been used, for example, for predicting the response of platelets to chemotherapy, and there it has been used in a more clinical setting. Let's put it like that. Still, the model was more complex than what we showed, but I think it's still a developing field, and it hasn't been applied too much. But we plan on applying it to more complex systems. The problem all the time is that it's difficult to find the people, because you need to have a certain hang to the dynamical models in order to confidently combine the model output with the machine learning part. So you need still skills in both, and there I still lack the people. But we set out a project, actually, or I sketched a project in the future. Let's see, with the Max Planck Institute, where I'm also associated, Max Planck Institute for Molecular Genetics. And there I sketched a project where we could try and use this informed machine learning for gene expression prediction. And gene expression is a very complex system where you have exactly many more molecules and where you have a high uncertainty in the models that you have, in the kinetic parameters that you have, in the dynamics that you have. And I'm really excited to see how this is advancing, how this is working out. But no results yet so far, sorry.
Hi, thanks a lot for your presentation. On the first problem, the way you tackle is cleverly using synthetic data based on the models. And do you see there is a gap that needs to be filled by crowdsourcing real data from the people? And if there would be a mechanism for people to crowdsource their own data, maybe there is like the ethical aspects, but if we take care of the privacy and the ownership of it for the people, incentivization, and as well as like hardware, like system on chip that comes into the hand of people, is there a need for that, or synthetic data is already good?
No, yeah, there is a dire need for that. So, because the drawback of synthetic data is that you have to assume that the mechanism that you assume in the model is true to a certain extent. And well, of course, if you have tons and tons of real-world data, they are to be preferred over synthetic data. So I think having more really measured data would enhance the field even more than using synthetic data. Yes, especially in fields like drug response prediction, for example, where there are systematic screens, but it's still not enough for the complexity that is being tackled there. So yes, it's starting also in Germany. It's starting like with electronic health records, with this Gesundheitskarte and stuff. So let's see. There's big potential for that. But in the meanwhile, let's take the synthetic data and try and prove a bit.
I have another question on the first problem, using synthetic data. And you alluded a little bit to this, that there's ways to put the physics directly into the model, for example, as a term and a loss function. And I seem to remember hearing a lecture here at TU on neural ODEs that there's ways to integrate this even more, to have a sort of more formal representation instead of learning from synthetic data, which can be inexact, right? Yeah, and I was just interested in your thoughts on integrating physics more in a more formal way into models.
So, yes, this is what we are currently also researching. It's at the edge of the research. What is the difference between using synthetic data, using physics-informed loss, using neural ODEs? And I think life sciences is more... It's a bit different than physics, because in physics you have these laws, right? I mean, the sand molecule is subject to gravity. That's not discussable. You can't discuss about that, right? It's the truth. In all the biological systems and biological models, a lot of uncertainty is coming. So this drawback that you do not include results or information correctly might not be as bad in biology than it is for physics, for example. And I think there we have a lot of potential. But I think we still have to explore. We have to explore this synthetic-based integration or synthetic data-based integration, this physics-informed formulation integration, and the neural ODE formulations. And they are all to be explored for life sciences. Yeah.
Okay, so if there are no further questions, I would like to give another round of applause for Katharina Baum for giving such a great presentation today. And now we basically open our second social break for the night. So outside we will have a few snacks, not too much, and a few drinks. So go around, talk to people. If you have any questions for the Flinter event, talk to this person in red over here. And if you want to contribute to the dataset, talk to me. And then see you hopefully on Monday. And if not, next Tuesday.